sequence. That's how we have those long chromosomes from, remember I showed you NCBI, we had the chromosome 1, and I kept scrolling, those hundreds of millions. That's how we actually put that together through a sequencer. So aside from that, I wanted you to know about restriction enzymes and microarrays. Restriction enzymes are enzymes which cut DNA at a very specific sequence. So there are different restriction enzymes recognize different sequences. So if we have on the board ATTCG, that restriction enzyme will always recognize that sequence and cut at that every occurrence of that sequence. So some genomes might have 100,000 occurrences of that sequence, whereas other genomes might have 10,000, right? So we can separate out all those different sequences using electrophoresis and see the sizes and the frequency of certain sequences. But more useful than that, we can add in genes to a genome. So if we want to cut a portion of the genome and then insert our own gene of interest, all we have to do is make sure that gene of interest has those sticky ends that's created by the restriction enzyme. So that's used as a tool. I used that example about insulin before. If you want bacteria to make human insulin, use a restriction enzyme to cut their genome, insert the insulin gene into their genome, and now you have a factory that makes insulin. That's bacteria.